So thank you very much for inviting me to this amazing venue, and as your speaker earlier said, please don't fall asleep during my talk. I know you've all had a really nice lunch, because I've had that really nice lunch too. So as Carla just mentioned, I'm here to update you on behalf of the steering committee of the British Association of Dermatologists Biologics Intervention Register, BADBIR. So I'm going to talk about why the study was undertaken, talk about the study aim and the design, update you on the progress, mention how the data is handled and used, provide some results to date, and also talk about future plans. So over the lunch, we were actually talking about the management of psoriasis, and when this study was conceived, the management of moderate to severe psoriasis consisted of topical therapy, which could be messy or difficult to apply. And if you saw a dermatologist, you would probably have availability of light therapy, which was quite inconvenient because might have necessitated many trips to the hospital. Also, maybe long episodes in hospital, and again, this was quite inconvenient, especially if you had a job and you would have to take that much time off work. And also, there was some systemic therapies, but these systemic therapies had recognized side effects, some troublesome, like nausea, fatigue, and perhaps others that were potentially more serious, like liver disease and renal impairment. So in 2005, the management of psoriasis was transformed by the development of biological therapy. And this is a protein-based therapy, and it's derived from living cells. So importantly, this targets specific parts of the immune system that is important in the development of psoriasis. And immune is a very important word in that comment. So why was this study undertaken? Because obviously these drugs went through the standard clinical trial process. And this would have been some evaluation in animal studies before, and also in the lab, before healthy individuals were exposed to a drug. And so there would be a study undertaken in healthy individuals to ensure that the drug was safe to be tried in patients. Once it goes into trials in patients, a small group of patients test the drug, and this is just to assess effectiveness, but also safety. If it proves to be successful at that phase 2 testing, it proceeds to phase 3. This is with a larger group of patients, and this larger group of patients, they also look for effectiveness, so how well the drug works, and they also monitor for side effects. And this can be up to, an important point here is this can be up to about 1,000 to 3,000 patients in total, and that's a large study. Eventually, it may go on to a phase 4 testing, so this will be where that drug is continued in those patients with whom it was successful for a longer period of time to monitor long-term side effects. So you could think, well, okay, that's been tested, surely that's safe, so why do we have to develop something like a BADBIR registry? These clinical trials have limitations. Firstly, they are run for a relatively short period of time. So in the context of psoriasis, which is a lifelong condition, patients are likely to be on therapy for most of their lives. These clinical trials, the patients may only have been exposed for a maximum of a year. So that's not very long in that context. There is a limited number of participants. So I said like maximum 3,000 patients. Psoriasis, there's 3% of the UK population roughly have got psoriasis, and this equates to, and please don't check the maths, in the region of about 2 million patients, of which about a third will have moderate to severe psoriasis. So that equates to about 660,000 patients. So when you think that these drugs have been tested in about 3,000, it's not particularly typical of the whole of the psoriasis population. Importantly, these clinical trials, if any of you have ever been a participant in them, they will exclude patients with some important other conditions. So this means that when the drug is launched onto the market, it's not really been tried in the real world situation. The typical patient in a standard clinic, it's not even been tried in a standard clinic situation, because obviously a clinical trial situation is one doctor, one nurse who you routinely see with a half-an-hour consultation. That's not typical of the way psoriasis is managed in an NHS clinic. So this is why BADBIR was established. It's an observational study. And this means that patients aren't put into categories dependent on the trial. The patient is prescribed the therapy, and then they go into the category, and they're followed up as they would be in a routine manner. Nothing special is undertaken because of the study. So the aim of the study was to investigate the long-term outcome of psoriasis patients treated with these newer therapies. Originally it was biologics, it's been extended recently, with particular reference to safety. And the main endpoints are cancer, infection requiring hospitalization, serious adverse events, and death. So if you remember, I mentioned that immune was a very important word in a previous statement. So the biologics, because they target the immune system, there is a theoretical risk that because the immune system is responsible for surveillance for cancer, that there could be an increased risk of cancer. Also, the immune system is responsible for activating the system to fight off infection, so there could be an increased risk of infection. So that's why those two were specifically pulled out. But then in addition, it was any other serious adverse events, because at the onset, you're never very sure exactly what you might find. So it was important to collect everything, and then obviously death. So the study is designed like this. Patients are assigned into a control group if they are starting one of the standard systemic therapies. And these are listed to the right of the slide. And they're assigned into the biologic group if they are starting one of the biologic therapies, and those are on the left, the numbers we're recruiting for at the moment, the drugs we're recruiting for at the moment. And these are the trade names of the drugs. And there's now a third small molecule group where we're recruiting some of the newer treatments, because these are not biologic therapies, and they've been entered into this group, Otezla and Skilarence. So for the purpose of the study, the events that occur are in each group are compared. So in the control group, if, for example, patients were seen to be developing myocardial infarctions, if there was a higher rate of events in the biologic cohort, you might think, oh, perhaps that is related to the biologic therapy, and that's what the analysis looks at. So patients are eligible if they've got a diagnosis of psoriasis, if they're under the care of a dermatologist, and if they provide written informed consent, because this is a research study. So the consent is taken in clinic. A healthcare professional explains the study to the patient and provides them with written information, and the patient has the opportunity to consider whether they'd like to take part in the study. Having read the information, if they are happy, then they will take part in the study, and the healthcare professional will start to collect some information. So this is the design of a study. The patients are followed up at a 6-monthly intervals for the first 3 years, and then annually until the end of the study. And we collect some clinical data, which I'll describe later. The patients also complete some information for the first 6 months, on a 6-monthly basis for the first 3 years. And also we link with Linkage Services, which I will describe later. It's on another slide. So the type of data that's collected, I mentioned about drug details. So we need to know about the drug details the patient's having for their psoriasis, because that's what assigns them to the various cohorts. We need to know about the psoriasis severity. We need to know about any other conditions and any treatment, any systemic treatment for any other conditions. And we also need to know over the period of time any adverse events or safety events that occur to the patient over that time. So for example, any serious infections, if the patient's had a myocardial infarction, any hospitalizations, all of those events are collected over that period of time. And the patients also provide information about their lifestyle. So for example, smoking and alcohol intake. And we are provided with information on cancers, deaths, and sometimes inpatient hospitalizations from the Linkage Services, but I'm going to go through that in more detail later. So the data is entered onto a web-based system, and this is transferred in an encrypted manner. So that's why that lock's there. So that means it's an unreadable manner that it can't be read by anyone who hasn't got the code. And then it's transferred to the University of Manchester. So how have things been going, you ask, or not? So it started in 2007. This is the pilot, really. It was to try and identify any problems there might be with data collection. So it started in a number of centres, some of the larger centres and some smaller centres. So it was officially launched in 2008. And a year later, the numbers of centres participating had doubled, and again doubled in 2010. And then it gradually increased over time until now it's 156. Most of the centres are in England, but that's because it's more populated, really. There's a lot more centres. Now, these centres, they are all hospitals, but sometimes there are one or two or three sometimes hospitals named within one centre, just because of the way the health service is structured, really. So this is how many it is, roughly speaking, in in England, Northern Ireland, Republic of Ireland, Scotland and Wales. And if I stay there long enough, you can count all the dots and see if it's all correct. But I won't. So as I mentioned, you can see how many centres were participating, and you can see then the registrations rose in line with the rise in the number of centres. And we now have just over 16,000 registrations. And a small number of these are children, 36. So we have over 10,000 patients in the Biologic group, 5,000, just over 5,000 patients in the control cohort, and 71 in the small molecule group. And that's because we've only just set this group up in the last couple of months. So just some milestones. As I mentioned, it started as a pilot in 2007, and it was officially launched in 2008. And at that time point, we were recruiting for these drugs, Enbrel, Humira, Raptiva, and Remicade. And Enbrel, Humira, and Remicade are all anti-TNF drugs, and those drugs were already being used in rheumatology. So there was some safety data already available on those drugs by the time we launched this registry. So interestingly, the following year, in June 2009, Raptiva, one of the drugs that we originally started with, was withdrawn from the market because of safety issues. And I think that's really important because at that time, that drug had gone through all of the clinical trial testing, and because it was only used in dermatology, there hadn't been an awful lot of safety data available. So it was only really when it's used in the long term that this quite serious safety concern arose, and therefore it was withdrawn. So in July 2009, BADBIR commenced registration of another one of the drugs, Stelara. And moving forward to March 2015, the 10,000 participant joined BADBIR. And in the same year, the first BADBIR study using BADBIR data was published. And also, at that time point, children were accepted to join the registry. So in 2016, 3 more drugs became available, and they also joined the registry. Cosentyx and Taltz were a new mechanism of action to the previous ones, but Benepali is what they call a biosimilar drug. So it is a biosimilar to Enbrel is Enbrel etanercept, and Benepali is a biosimilar of etanercept. And this is a new development that's increasingly being seen now. 2017, the first paper on safety data was published. And in 2018, Erelzi joined the registry, and that is another biosimilar of etanercept. And these other 2 drugs, which are tablet preparations, Skilarence and Otezla, and they form the new cohort. So now a mention of the BADBIR team. The data is handled primarily in Manchester by the BADBIR team. and this is the BADBIR team, or some members of them. The BADBIR bear, who's with me here, holding my hand, he's at the centre of this BADBIR team, and he's actually blocking me out that picture because I was having a particularly bad hair day, and I thought the bear was definitely an improvement on me. So as I mentioned, the data comes from all the hospitals via the healthcare professionals in an encrypted manner, and is entered into the database. So the BADBIR team ensure compliance with research governance regulations, because there are quite a lot of research governance regulations, and it's important that we comply with all the rules in that respect. They make sure the data is kept secure. They check that the data is accurate and complete, and if there are any inconsistencies, they will query that with the centres to try and ensure that there's accuracy. And then they also provide support to the hospitals, because sometimes there's a training of staff needed all the time, because there's new staff continually recruiting and collecting data for the study, and also sometimes they may have a query on eligibility, and so there's a telephone helpline to help them, or else they also have a email correspondence. So the data then, once it's captured on the database, is used in three main ways, and these are the three main ways, and I'm going to talk about each one in turn. So the first one is some data on safety is transferred to the drug companies that are participating in the registry. And this, again, is non-identifiable data, and it is used by these drug companies to report to the organisations that collate the safety for the governments of the various agencies. So that may be in the UK, that's the MHRA. So these pharmaceutical companies at every year have to develop a safety report for these agencies, and this information is collated so that they can be sure of the safety of the drug. And in a way, that was what happened with Raptiva, as I've mentioned earlier. Reports came through to these agencies, and they became concerned about that drug, and then they asked for it to be withdrawn, which it was. So this is how BADBIR contributes to that, and that's how it fulfills the requirements for the companies for reporting safety. So now linkage. So I mentioned linkage earlier. So we transfer identifiable information to the linkage services. And you may say, what are the linkage services? Well, these are databases that are embedded in the healthcare systems within the NHS or in Ireland also. And they provide information on cancers or deaths and inpatient hospitalizations in some cases. And you may then wonder, well, why are you not getting that from the hospitals? You're already getting that information from hospitals. But sometimes patients don't turn up at the hospitals. They decide, I've had enough, I'm clear, I don't need to go back. Or something untoward happens and they don't go back. And in a study like this, you could draw different conclusions. So if you drew the conclusion that you didn't have any more information on that patient, so it was a bad outcome, you could make the safety record of that drug quite bad by including that. Or you could go the other way and say, oh, actually, we haven't had any more information on that patient, so it must be great. You could make the safety record of the drug very positive then. So in order to ensure that we don't do that, we collect information, so even if patients don't come back to the hospitals, we get this information. And obviously these are quite important serious events that we do need to capture. So that makes sure that the data is completely comprehensive, and we can be more robust, and we can be more confident about the results. And finally, but importantly, it does get transferred to researchers. So the researchers, the steering committee and the researchers decide on an outcome that they should explore. The researchers undertake the analysis, and then the results are published. So now I'm going to talk a little bit about some of the papers that have recently been published. And I'm going to concentrate on two areas. One is the drug effective at treating psoriasis? And the other area is the drug safe long term. So 3 papers have been published related to effectiveness. And the first two were looking at drug survival. So if you stay on a drug for a long time, it's likely that the drug is either working for you, so is therefore effective, or you're able to tolerate the drug, so it's therefore hasn't got as many side effects, lack of side effects. So this is what was explored in the first two papers, which I'm going to show pretty much side by side, really. So the first paper looked at first-line biologics, so basically the first time a patient had been put on a biologic, their first exposure. And the drugs that were explored were Humira, Enbrel, Remicade, and Stelara. And the results were that drug survival decreased over time, so from 77% in the first year to 53% in year 3. But there was differences between the drugs. And there were various suggestions as to why patients might discontinue, and there were other suggestions as to why they would continue. And Stelara had the highest survival rate, and this was followed by Humira. And the next paper looked at what happened if patients switched from a first biologic onto a second one. And the drugs that were looked at here were Humira, Enbrel, and Stelara. And the results were very similar because they also found that drug survival decreased over time. Again, 77% at year 1 and 58% at year 3. Again, there were some predictors suggested related to drug discontinuation and also predictors for drug survival. But the important point, I think, from this and the sort of take-home message is that in my experience, not too distant past, there were some centres where they weren't allowed to try patients on another biologic. And I think that this does emphasise that if a patient has not benefited from the first biologic, that the second biologic is also likely to be effective. So there's evidence here that it is important to try a patient on the next biologic. Again, Stelara had the highest survival rate here. So this last paper related to effectiveness explored how the biologics impacted on health-related quality of life. And this is the patient perspective on how the biologic had improved life for them, as opposed to an assessment of their skin or how long they've stayed on the therapy. This was what they felt, whether it improved or not. And it was assessed using a DLQI score. I don't know if any of you come across the DLQI, but it is a questionnaire that's administered to patients. And the questionnaire evaluates, the patient completes how they feel the psoriasis is in fact affecting their lives. And if you have a score of 0 or 1, there's very minimal impact on your quality of life. So the findings were that nearly 50% of patients scored 0 or 1 at 6 months, and this was sustained at 12 months. And more than 80% had a change, an improvement in their quality of life. So again, this shows that the biologic therapies were effective in terms of the patient's perspective, but they also found that although the biologic therapies improved quality of life, there were some other issues that might influence that. Firstly, the choice of biologic, also some lifestyle characteristics such as obesity and smoking, and some comorbidities. So I guess the suggestion was that you could possibly influence some of these areas. For example, quit smoking and lose some weight, which might improve the effectiveness of the biologic therapy. So moving on to drug safety. This is the only paper that's out there in the public domain on safety at the moment. And this one explored the risk of serious infection. Again, it was Humira, Enbrel, and Stelara that were investigated. And the results were that they did not find a significantly higher risk in any of the biologic therapies compared to the control cohort, because as I mentioned, we always have that control cohort to compare against. And also, there wasn't any significant difference in the risk of serious infections occurrence across the 3 therapies. So this is important because in the clinic, the risk of serious infection doesn't need to be accounted for when you're considering which biologic therapy to start or whether to start a biologic or not. This is what this is suggestive of. So this slide illustrates that we started in the clinic with the healthcare professional and the patient. The patient decided to take part in BADBIR. The data was that particular patient's data was transferred to the BADBIR database. It was combined with all of the rest of the data, combined with the data that we also acquire via the Linkage Services. The researchers analyse the data, publish the results, and then this feeds back to the patient in the clinic, where the healthcare professional can now discuss those findings and how those findings refer to his or her particular care, and that this is how it helps to further the knowledge. So upcoming publications, there's an analysis underway at the moment looking at the rates of cardiac events, such as myocardial infarction, etc. They are also exploring the risk of post-surgery. I think it has actually post-surgery infections as opposed to complications in general. It's expected that, in fact, it's being presented very soon at various conferences, the rates of non-melanoma skin cancer in the registry, and these are basal cell carcinoma and squamous cell carcinoma. There is a paper about to be published on the rates of cancer, solid tumour cancer, liver cancer, lung cancer, etc. And this is in collaboration with SONET, which is a group of other registries in Europe. Further down the line, but something that we are exploring, is the risk of cancer in the registry, in our population, in the BADBIR registry, and also going to try to look at pregnancy outcomes. Because when I talked earlier about clinical trials, obviously pregnant patients are not included in clinical trials, but patients do get pregnant on therapy. And so this is an excellent opportunity to just review what has actually happened in those patients who have often accidentally got pregnant. And if we can report on that, we won't have lots of them, but by joining it with perhaps other evidence, it might help to support that patient in the clinic who is making a decision on whether or not to become pregnant when they are taking these therapies. So these are references. These are on the BADBIR website. There is a publications tab, and you can go on there and have a look through these, read the abstracts, and often get to the full text. BADBIR also collaborates with other groups that are undertaking research in psoriasis, such as the SORT group, the BSTOP group, also IMAP, which is looking at medication adherence, and as I mentioned, SONET. So this is to try and maximise the benefit of this data to try and answer as many research questions as possible. So future study plans, this is really about the study itself and not about the research plans. So the study end date has been extended to 2028. We've got ethical approval to continue to that time point. There are, maybe you know, lots of new drugs, lots of biologic drugs about to be launched probably in the next year, and all of those companies are in discussions with the BAD to join the registry, so that's really good news. We want to extend on the data we've got on children, because the children are exposed at quite a young age to maybe these systemic therapies and possibly will be on them for a lifelong period. So it's a very important group that we want to try and collect more information on. Although we do expect that the numbers are still likely to be quite small. And we are also in the process of developing a patient app. And this will be at the onset at least, where patients can complete their questionnaires without writing them on paper first. We feel that probably the time is right to do this. So this is our website. There is a section on there for participants, but it is open. You don't need to, you can just log in and find that section. And there's information on there. Newsletters are on there, patient newsletters. And it includes summaries of the research papers. We try to get that in there as well. So feel free, we have some of those here. So if you want to have a look at them or you want to go on the website, we're very happy to take some recommendations on how these could be improved. That would be excellent. So I want to acknowledge that the British Association of Dermatologists is provided with restricted income financial support from these companies. The BAD, within the governance of this study, which I didn't mention, what I will now, there is a steering committee, and they oversee all the activities of the registry. These are all either British Association of Dermatologists Association members or BDNG members. The BAD then commissioned the University of Manchester to set up BADBIR, and so the actual study is run from the University of Manchester using the financial support that has been provided by the companies. The companies do not have any impact on any of the research. It's completely independent from the companies. They get an opportunity to comment on it, but they can't prevent it from being published. So some thank yous, especially to the patients without whom we would not have this data, to the hospital research teams who work really hard to provide the data, and also would like to tell you that we have a Twitter account. I have absolutely nothing to do with it. I have no idea how it works. But those young people in my office, they sort all that out. So we are tweeting that we are here today. So thank you all, especially for listening to me. And I didn't see anyone fall asleep, but I probably wasn't looking. That concludes.